Yeah, thanks. As Elisha mentioned, Kathleen and I are both on Pacific time, so bear with us here. You can watch the sunrise through the window behind me. Um, okay, is this sharing correctly? Okay, perfect. Okay, thank you. Yeah, so Kathleen and I today are going to talk about qSIP analysis, kind of broadly speaking, and then how we have chosen to do a kind of a first pass incorporation into KBase, and this is where we're looking forward to some community feedback about the best way that would make it the most useful to users. Sorry, there's about 12 Zoom windows I need to minimize and move out of the way. Okay, so first of all, a little background. This work is a collaboration between KBase and the Microbes Persist SFA at Lawrence Livermore. The Microbes Persist SFA is interested in connecting microbial activity to soil carbon persistence. So we use a lot of stable isotopes to try to track actually where carbon is flowing in an environment. Other work at Livermore works with tracking nitrogen. We image isotopes. We use a lot of nanosims. So we're generally interested in how we can use isotopes to help actually explain metabolic activity, the flow of nutrients in systems. So a little background about SIP in general. So a lot of biomass in, say, a soil sample is actually inactive. So inactive can mean it's not growing. It can also mean it's long dead. It's just organic matter that's stuck to minerals or kind of floating around in a marine system. But this data typically still shows up when we do various omics analysis. So we use the stable isotope probing to add some sort of tracer elements, and then any incorporation that we see in biomass, we can say, okay, that's new biomass. It's something that's actually produced since we added that label. And you guys, I'm not sure if you can see my mouse, but I'll assume you can. So this is just a schematic from an upcoming paper that we have that's currently on bioRxiv that details both SIP, qSIP, and generally these standards that we're trying to develop called MySIP. So you'll hear the term MySIP for minimal information about a stable isotope probing experiment. So the schematic just on the left, you add some sort of isotopolog, and an isotopolog is just a labeled, a heavy labeled version. It can be typically carbon C13, nitrogen 15, or oxygen 18, 18O. You do some sort of incubation. In this situation, we're extracting nucleic acids. You spin down the nucleic acids, and those sequences that incorporated more of this heavy label will centrifuge to a higher density in the centrifugation, and then we can separate these by taking out different fractions and sequencing the different fractions. So traditional SIP, you just kind of have some cutoff and you say, okay, everything above a certain density we're going to say is active. But that kind of limits the analysis, because if you're working with DNA, for example, you have GC content. So some microbes, some DNA content is already going to be heavy. So you might be mislabeling a high GC microbe as active just because it does naturally centrifuge down to those higher densities. Similarly, you might have low GC content organisms that, even though they're taking up substrate, they might not actually be heavy enough to come into that, whatever it is that you labeled as an active portion. So we use quantitative stable isotope probing, or we'll actually have a paired sample system where we have an isotope log of natural abundance, and then it's paired with some sort of isotopically enriched. So in this situation, we actually have two sets of triplicates. And then we can see, okay, what is, if this is an amplicon-based sample, for example, at what density does the natural abundance sequence run? And then at what density does the isotopically enriched run? And we can use those differences in density to try to calculate an amount of isotopic enrichment. Further, we can use this not just for labeling of a specific substrate, but we can actually add general substrates like 18O water and do a global population estimate of growth and mortality. Okay, so there's a couple terms that we use. So the kind of the metadata around qSIP can be kind of complicated. There's a lot of steps, and from your original sample to fractionations to kind of downstream feature selection. So there's kind of three levels of data. So the first is the source material. So this is your, your plant, your scoop of soil, whatever it is that you actually added your label to and extracted DNA from. And that source material is fractionated into samples. So the sample is the middle level where if you traditionally for qSIP, we sequence with, Illumina. So this is like your what you actually ran in the machine. So each sample has, a density associated with it and also some DNA concentration. So this is post-fractionation. How much DNA did you get out of that fraction? And you can also calculate that with qPCR if it's amplicon based. So the more fractions or samples, the more accurate you can actually determine what that weighted average density is. But more samples is also expensive. So there's kind of a give and take with how many samples you actually want to sequence. So typically what we see is for an ideal sample curve is we'll see a small shift. So this is a 12C in blue and a 13C shown in red. So kind of the peak is similar, showing most bugs are not actually active. Most are taking up more 13C. But there is this little hump right here that shows, okay, there are actually some DNA sequences that are migrating further in the density gradient. Okay, and then so the samples, the individual fractions are then what are sequenced to get what we're calling feature abundance. So there's three levels, yeah, again, just source material, samples, and then now features. So each feature could be associated with an OTU, an ASV, a contig if you're doing metagenome sequencing. So each has its own weighted average density, both in the light unlabeled isotope and then the heavy labeled isotope. And once we associate the weights based on their abundance, again, with qPCR counts or DNA concentration, we can then at an individual feature level determine this shift in weighted average density. So just showing two different examples here. This ASV2 kind of has a clear visual shift in abundance or in isotopic enrichment, whereas ASV9 doesn't look like it's taking up additional heavy substrate. So the qSIP process itself, how do we actually turn this from a visual, yes, it looks like it shifted to the right, to an actual quantitative component? So starting first with the 12C in this case, or sorry, this is a 16O study, 18O study. We can first calculate out the molecular weight of the DNA at this density. So we start with the weighted average density, convert that to GC content, and then from GC content, we can calculate a molecular weight. So for this individual ASV, the weighted average density was about 1.72, and that comes out to a molecular weight of about 308. So next, what we can do is, since this is an 18O study, all nucleotide pairs, whether they're ATs or GCs, have 12 oxygens, or sorry, have 6 oxygens per pair. Since we're adding 2 additional neutrons, we're going from 16O to 18O, we can then assume that a 100% labeled microbe, in this case, would have a weight of 308 plus 12. So that's, again, 6 oxygen atoms times 2. So a 100% labeled sample would have a molecular weight of about 320. So then once we add on, what did we actually see, and get its weighted average density, we can see it's about 313, which is about 4/10 of the way up, or about an EAF value of 0.398. So EAF is the term we use. It's the excess atom fraction. You may have heard AFE. It's the same thing. So this is how we can go from, okay, this particular microbe, this particular feature in this case, which happens to be an ASV, is about 40% enriched. And again, really, that's just saying of the oxygens in the DNA, about 40% of them appear to be 18O. So these EAF values can be compared between treatments. So one EAF value is still within this paired sample. So if you wanted to compare something like normal versus drought, you would come up with, you would have to come up with really 12 samples, a paired sample for the normal and then a paired sample for the drought. And then you can show them next to each other. So this is just looking at a random set of 25 features from this study.
So since this is a workshop about qSIP analysis in KBase, we wanted to take a moment to briefly orient you in KBase in case you don't already use it. If you are a KBase power user, just sit tight. And everybody in the audience can get out your fair bingo card for this part. So the systems biology knowledge base provides access to biological data as well as bioinformatics tools for analyzing that data. In KBase, we create shareable and, if you'd like, citable records of our work called narratives. And so over here, I have a little toy drawing of a simplified narrative. And one icon that I've emphasized here, it won't be as big when we get into the actual narrative, but this is really important because this is how you control sharing your data. So by default, your data, including any SIP data you bring into KBase, will be private at first, but by clicking on this, you'll be able to control sharing that with specific collaborators, with a whole group of collaborators in a group called an org, or making it public for everyone to see. And you can talk to the KBase team about getting a DOI assigned and making a citable narrative to go along with any publications. So in KBase, your genetic sequences, your sample metadata, other types of measurements are going to be uploaded into interoperable data types. I'm using interoperable in a narrow sense here, in that if you upload your data as a genome type, for example, that will then work in all of the apps that accept a genome in KBase. And so when we get into the narrative, there will be a collapsible pane, and over here on the upper left, this is where you will go to see all of the data that's present, all of the different types of data that are present in your narrative. And then this main section of the narrative over here is composed of 2 different types of cells. The first type is markdown cells. So this is where you can write and add links and perhaps some figures to give context to your data. And then the cells that are actually doing the work of running the bioinformatics software here will be the app cells. And if you're curious about what types of apps are available, you can look at the app catalog, or in any narrative, you can come over here. You can browse the tools that are available by category, or you can search for a specific tool. One thing I want to note today is that because the qSIP tool is under development, you won't be able to search for that on KBase right now. It's on a separate site. So that will be coming, but just know that you can't go on there and search for the qSIP app today. And so when we work in these apps, you'll notice us using 2 tabs today. The first is we'll be showing you a configure tab, and this is where you can come and see a drop-down menu that allows you to select the types of data in your narrative that you want to run the analysis on. And then once the job has finished running, you'll be able to come over to a result tab and view the output. So I think that's the only other thing I wanted to note was that all of the apps and your narratives themselves are versioned, and so this helps to make the workflow reproducible. So that's your very quick introduction to KBase. One great thing about KBase is that it's extensible, so it provides templates and support to help developers like Jeff add new workflows like the qSIP analysis into KBase. And this allows us to be responsive to emerging analysis needs. For example, the need for tools to work with the growing body of sequence data that are generated by stable isotope probing experiments. So over here, we have a figure with time on the x-axis, and then on the y-axis is just a number of publications with some key search terms related to stable isotope probing that link to sequencing data in the sequence read archive. And you can see a very clear trend here. There's a marker here for the introduction of qSIP. And one landmark that's not on this plot towards the end, but probably should be, is the introduction of an experimental pipeline for qSIP at JGI, which will certainly increase the accumulation of this type of data in the community by making the ability to produce it more accessible. All right, so I'm going to talk about bringing the qSIP2 workflow into KBase. But first, I wanted to step back just a moment and say a few things about what the motivation is for doing this, why we're bringing it into KBase. One reason is the support in KBase for data sharing and reuse. So we started this with kind of the motivating scientific interest of how microbial functional roles relate to element cycling in ecosystems. And having a standard tool for running this analysis can support combining those results downstream in meta-studies so that we can learn more about how microbes contribute to and transform or stabilize organic matter across different sites. Another great reason to do this in KBase is the transparency in data analysis. And so, for example, we're interested, as Jeff mentioned, in the shift of that weighted average density for a taxon between unlabeled and labeled treatments. And that calculation can only really be done if that taxon, that feature, that sequence feature is present in enough fractions of the density gradient, and if it's present in enough replicates of labeled and unlabeled treatments. And when I say enough there, that's obviously one of those points in a workflow that represents a decision, and that decision will impact how you can synthesize data downstream. So it's really great to do that in a narrative environment where someone can easily just copy over your analysis and rerun it with perhaps some different filtering parameters. And then the final reason is that KBase can support integrating qSIP results with other types of data. So since qSIP kind of sits at this interface of biogeochemical data and genetic traits, we might want to link measures of how a microbe is using substrates that we get from qSIP to other types of data. amplicons, metagenomes, chemical abundances that we can bring into KBase. So that's a little bit about why we're doing this. As far as the code we're bringing into KBase, that is hosted here. It's this qSIP2 package in R, and Jeff has done a lot of really wonderful work to refactor and document this. I will just say, as someone who is very new to this type of data, that I found it very easy to get started with this, and it has actually taught me a lot about how this type of data works. So it's awesome. And so today we're going to discuss the data that are generated by a qSIP workflow and demonstrate how to upload that into a narrative. And then we're going to demonstrate how you run this qSIP2 workflow from the R package in KBase. So you'll have access to all of these functions that run all of the parts of a qSIP workflow here, creating a qSIP object, comparison grouping, filtering your features, resampling, calculating that excess atom fraction through a KBase app. However, we're also going to need something from you after we present all of that, which is some feedback on how all of this is working. So I have some discussion prompts here. I'm going to take a minute to go through them right now, just to prime your brains for feedback before we get into the demonstration, so that you can be thinking about what your answers to these questions might be, or what other questions you have. So we're interested in what kinds of experimental designs the apps will need to accommodate. How many different types of treatments? Are you doing things that involve a time series, that type of thing? We are interested in what types of metadata on experimental or environmental sources and from the data processing are needed for you to reuse and compare data. We're going to discuss the metadata that we are collecting, but we'd like to know what else you feel is needed. We want to hear what kinds of new qSIP modifications might be on the frontier and might require the apps to adapt in terms of what they take in and are able to put out. And we want to know what kinds of genetic data you might be using with this, because this qSIP workflow can accommodate both metagenome and amplicon. And if it is amplicon, what types of marker genes might you be using? And finally, we'd like to hear a little bit about, once you see this in the demo, where would you like maybe more or less modularity or more or less flexibility in how you set the parameters in order to make this work for you. Okay. So before we get into the demo and start importing the data, I want to talk a little bit about the type of data generated in a qSIP experiment. So we're going to walk through once more that wet lab procedure that Jeff introduced, and we're going to do it with a mind towards what types of data we're generating at each stage here. One theme I kind of took from the BER data infrastructure session yesterday was just the complexity of biological data sets that we generate and how this can challenge our metadata collection and standards. And I think qSIP is a really great example of this because it creates metadata with different levels. There's a hierarchy to the metadata, which we're going to see here. So you have your source materials to which you introduce one of the isotopologues, either the isotopically labeled or the natural abundance compound. You have these in replicate and you incubate them. You're then going to extract nucleic acids. And so there's some standard metadata through this part that is covered by the MySIP standard that Jeff mentioned. And I won't get more into that right now, but we have some more slides on it if you have questions about it at the end. There's also likely some user-specific metadata related to whatever your experiment or study was that you got this source material from. And so then you're going to take some measure of the nucleic acid content at this source level, whether that's concentration or a marker gene count by qPCR. And then you'll use the ultracentrifuge to stratify the nucleic acids in cesium chloride into regions of uniform density. And so at this point, you're going to have metadata for each of these processes that are associated with those sources. And they're labeled samples here. We'll get into that. All right, so there's metadata that's generated at this source level. So then you're going to separate that material into density fractions from heaviest to lightest. You're again going to take some measure of the nucleic acid concentration or qPCR count for each of these, and you're going to record the position of each fraction in the density gradient. So this is another set of metadata that's being generated at this level. So you can think of these as like parent and child or sample and subsample. We're using that qSIP2 language of source and sample here. You could also say pre-fractionation and post-fractionation, whatever works to keep it straight in your head. We're generating 2 levels of metadata here. And then, of course, the features that we're calling from sequencing. And here I have this as an amplicon workflow. And so this brings up the question of what to call these different levels of metadata and how to keep track of those as we move through a qSIP workflow. So when you bring the data into qSIP, you're going to have your source data and your sample data, and those are going to be linked by a field that we borrowed from the minimal information for any sequence standard from the Genomic Standards Consortium called the source material ID. And so in your sample data, you're going to assign each sample, each fraction here, a source material ID that links it to the name of a source that it came from. And so that's how this linking between kind of the parent level and the child level is accomplished. And then each name for the distinct sample that's linked back to a source will have to match a column in that feature ID table, in that feature table that has the counts for each of your amplicons. And so that linking will just be accomplished by the sample ID name. When these data are brought into qSIP, each of them comes through an import function to become a qSIP2 object, and then these are put together into this. These are put together into kind of this master qSIP2 object that the analysis is going to run on. And the reason for this structure is that it allows us at this stage, when everything is coming in, to do some validation for how the data is formatted, to make sure that you have this linking between the source data and the sample data, to make sure that you're using an accepted isotope pair for the experiment, and to make sure that you've met some of those MiCIP metadata requirements. So when we bring this into KBase, we have, for a couple pieces of it, a fairly straightforward mapping of how that metadata is going to be recorded. So for the source data, we're going to use a KBase sample set. In KBase, we call it a sample if it's that environmental material to which you have different metadata on your collection. And then for the feature data, that's going to come in as part of an amplicon matrix. An amplicon matrix is created by uploading a table that has counts for each of your sequence features in each sample, together with the sequences associated with those features. All right, and so we have the source and the feature data. All we're lacking is kind of that sample level. And so this is just going to be accomplished by uploading a second sample set that's going to contain source material IDs that link back to the source data. So that's pretty straightforward, and you can run your qSIP analysis from there. I'm going to skip over this part because we're already a little behind, but this was just to kind of emphasize how the number of samples that you have to keep track of can grow depending on whether you're just looking at one environment or whether you start adding more environments or more conditions. It becomes a lot to keep track of. And so there are, and we want to stay focused on the fact that we've got to compare, like Jeff said, the unlabeled and the labeled samples within a condition before we can do any analysis with those excess atom fractions to compare between condition 1 and condition 2 here. And so in order to help us keep track of which samples we want to compare, in the R package, there are a couple functions to do that. In KBase, we're going to have the aid of what we call a data combinatrix, and that's going to be introduced in the demo. So with that, I think we're ready to get into the demo finally. All right, so this is an introduction, a little introduction and background to qSIP2 in KBase. Giving shout outs to Jeff and to AJ, who's on the call. She created the combinatrix and that we're going to see today, and just giving a little background on qSIP and some of the data requirements. So in this first part of the narrative here, we're going to import the qSIP source data. So for this, we're going to use the import samples app. And so to bring in my comma separated variable or tab separated variable file that has a row for each source and a column for every field of metadata that I have at that source level, I would come over to data here to import, and then I just dragged and dropped the files that I'm going to use today, which are here from my computer. And they loaded up here in my staging area. And so when I open up this app to import samples, the drop-down menu here will let me see the files in my staging area. Here I select my sources table, a template format. There's some more detail on this in the KBase documentation, but we're just using a basic KBase template. And then I can write some detail to help me remember where this data came from before I uploaded it, what it is. And so once I've uploaded these sources, I get this summary here. So there are 10 sources in this data set, and I can view what I've uploaded here. So each source has a unique name within this data set. Ideally, you would probably get a global unique identifier for samples in a large project that you'd like to upload here. And then we have some of the MySIP metadata fields indicating whether each source was treated with a natural abundance isotope log or one labeled with a heavy isotope. It's got some measurement data, so I can kind of check all of that and make sure it uploaded as I wanted. This app cell here was just used to update the access controls to my samples so that Jeff could use them. And then the second step here is I use that same app. So it's just a repeat of the same thing to upload a second table, which is those fractions. And so these are the names of each fractions. Here in this example, it's just related to the they're just given sample and then a number, but you can see over here that source material ID column where it tells you which source they came from. And then I have my fraction associated metadata. And then the final stage of upload is to import that amplicon matrix. Sorry, I've got to move this around the Zoom window to show you the configure. So here I selected the abundance table with the amplicon counts. I'm going to have a FASTA file with the sequences for those counts. There's some metadata collected about which marker gene you were sequencing, which region of that marker gene, your sequencing platform, and just broadly the bioinformatic approach you used to call amplicons. And so you fill that out. You run the import app, and then we're all set to do KBase. We can see over here in the data window, we have the sources, the samples, and our amplicon matrix.
So starting where Kathleen left off, so the qSIP R package is fairly mature, I would say. The KBase portion is as Kathleen mentioned, very much a beta, even alpha run. So we're trying to figure out all the best ways to take the more complicated R package and distill it down to a nice app. So I'll first start with this combinatrix. So a qSIP analysis requires you to define a couple things in your metadata, which column, for example, contains the quantitative part, the qPCR counts or the DNA concentrations. Which column has the metadata that you want to use to pair and say, okay, this is the set of 16.0 I want to compare to this set of 18.0. It doesn't always make sense to just run all possible comparisons. So we want to make a way that you can define what it is you want to compare. So a great start for this is to look at this combinatrix and where here the sources, samples, and amplicons are paired how they are. So each row basically is this taxon IDs, this is its value in this sample in this source. But we can click on this and expand this a little bit. So I'm just doing this to kind of show that I want to get the column names when I actually run the qSIP app. So the first thing I'm going to look at is this column called time. So this data set has 5 16.0 and 5 18.0 samples all at day 10, time 10. So I'm going to use this time column to say, okay, these are I want to do EAF values at day 10. And then the second column I need here for now is in the samples, this qPCR 16S copies per gram of soil. This is my quantitative part. This is saying, okay, in this fraction, normalized back to per gram of soil, this is how many 16S copies that I got. Everything else, luckily for our situation, these other column names, we will need them, but they all already conform to the MySIP standards. So for example, the gradient position density. So this is the density of that certain fraction of gradient of fraction 1. So we'll need this data as well, but the qSIP app will take these default MySIP, will take these MySIP names by default. So if your data already has column names that are MySIP compatible, then there's no need to change anything. Okay, so coming here now to the actual qSIP app. So we start off, this has already been run, but we have our source data, sample data, and feature data. So if this was I don't want to rerun it just so since it's already been run, this will be a drop down and show all of this sample data. So this source drop down would show either the sample or source, since those are both sample sets. Same with this sample data. And then this feature data would only show this amplicon matrix. So you just select your 3 objects that you're interested in. Then we go down to this next step. These are asking for column headers, as I mentioned before. So since the source isotope column, isotopolog column are already named according to MySIP standards, the pre-filled name will be fine. We won't need to adjust it. Here's where I say the run group column. If you click this little question mark over it, it'll cover the little short description. So this is the column header that groups the source material into different qSIP runs. So as I mentioned, we want to use time for that. Source mat ID here, this is saying, okay, in my sample data, which column has the source material ID? So this is how I'm linking sample data to the source metadata. Gradient position and density are already standard. And then here, this total abundance, I copied over this column that I mentioned. Internally, the qSIP app will normalize your data according to this. If you already have done that in your data, you can give us can give a column name. Otherwise, you can click this button to say instead calculate that relative abundance, and it'll take all of the data in your features and normalize it to the amount of qPCR copies or DNA that you got in that fraction. And then the last of the main is your feature data. What do the counts actually represent? Are they, is it raw counts from Illumina? Is it coverage? Say these are mags, and you've normalized your data to read depth per nucleotide. This data is relative, meaning everything's already been normalized to 1. It's all compositional data. And this is really just those validation checks that Kathleen mentioned. If you say counts and it's expecting integers, and you'll get an error if you give floats or some non-integer data. There are some advanced parameters. If we click this, these are filled in by default. So the qSIP, the analysis workflow does this bootstrapping resampling step. So this is just how many bootstraps do you want to do. We can allow for some of the bootstraps to fail. I don't have time to go into that right now, but you can set this, okay, resample success rate. I want at least 80% of the resamples to succeed. And then your confidence level. So after this bootstrapping procedure, basically how big do I want my confidence levels to be? Okay. So like I said, this has already been pre-run. It took less than 2 minutes to run, which is great. And the result for now, there's not a lot of reporting that's happening yet. Rather, some plots and some summaries are displayed just as links right now. We plan to build out the reporting of this, and could use some user feedback about what you might want contained in that report. But we can see some things like, okay, some filtering plots. So based on the sources, how many of the features actually passed different filtering. So this is actually a pretty dense data set. It's not very sparse. Other data sets you might find, you have features that just don't exist in some samples. That's not necessarily the case with this one. The EAF plot, so this is across some 300 samples, the zeros here. With this 18O study, we could see that a lot of the features shown on the y-axis have some degree of enrichment, have some degree of growth. Some have more than others, and some don't appear to be growing. So just things like that. Here's an actual summary table with EAF values and some different statistics, confidence intervals, how many resamples succeeded, failed. So yeah, like Kathleen mentioned, this is still in app dev. It's in development. It's not in beta yet, but hopefully we'll get this fleshed out and make it available soon.
So maybe, Kathleen, do you want to put the slide up with the question prompts? So one of the challenges, one of the blessings and curses, I guess, of KBase is that we have a graphical user interface that allows people to sort of visualize what they want to do some drag and drop bioinformatics for some pretty complex analyses. And so the goal for taking the R package and putting it into KBase is that you don't necessarily need to know how to run qSIP on command line in order to do qSIP analyses. The challenge is there's a lot of variables, as you see, when Jeff is building out the user interface for what comparisons you want to make, how to identify those. And so what we're looking for as people get comfortable and start to use qSIP more is to understand what that user experience in that drop-down menu needs to look like to make it intuitive. And so if people have comments now, that's great. We'd love to take them. There's also a Q&A doc I shared with the online Zoom community, but we can share it out with everyone here after as well. We'd love to get feedback from you on what kinds of comparisons you're looking to do, what kind of metadata are you thinking about collecting or you have collected related to qSIP, and then how do you want to visualize the data and the results after so that you can determine how effective the qSIP analysis was. Okay, so I'll pause there. Does anyone online or in the room have thoughts? How many people have done qSIP before? You have a question? You have done qSIP? Okay, do you mind going to the microphone? Okay, perfect.
All right, thank you. That was a great talk. So sometimes we see variation between spins and between tubes when we do qSIP. So I'm just curious and, would encourage maybe if there was a way to adjust for that. We usually, because we want to compare across different tubes and spins, if there's a way to compare for or adjust for that variation within the app, because we do that manually in R right now.
Yeah, I would love to chat with you. We have some methods to try to determine what that shift in density might need to be. Going forward, we're starting to see more, and this is what if you use do qSIP through JGI, they've really standardized this, is the introduction of actual spike in standards. So this is a known GC content. We know what density it should come to. So then you can more empirically shift your densities later. But yeah, like I said, we have some methods that we've used in the past. If you have a novel method, definitely let me know, and we can see about putting that into the R package, and then making it maybe a drop down in the KBase app eventually. Like I want to correct my densities using this method. Yeah, but it's not built in currently, but I understand the need for it.
Yeah, that's a perfect feature request. Like that's what we need to hear from people that they want to have more control over. So let's make sure that you get my contact information and we'll set up a chat. Cool. Any other thoughts or questions around this? So Jeff also is working actively with the JGI staff as well. They are releasing quantitative probing analysis for metagenomes. And so we will be adapting this to the JGI outputs and to some of their, like you said, he spikes in standards. And so we'll have to adjust for that as well. So this is an evolving process. And if you want to get your experimental requests in now's the time to do it. Okay, if there's no other questions, we'll go ahead and wrap up. All right, thanks everyone.
Thank you.
And thanks to Jeff and Kathleen and AJ for all the work putting this in for the workshop. So thank you.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.